in this example, I'm asking you to match the left side to the right side. So the last answer is the correct one. So good job, guys. And let me explain the figure so that you're like, oh, that's how, if you didn't go by elimination. Okay, so if you change the coding sequence of a gene, maybe it will affect the phenotype, maybe not, you will learn about silent mutations. Or if you change the transposon, so regulatory element is the promoter of a sequence, and sometimes you get transposons landing the promoter, but okay, regulatory element means DNA, regulatory element means the DNA is regulating something, so it's the promoter, it's regulating transcription. And I'm saying I'm not changing the coding, so coding is the exon. That's what the sequence of DNA that ultimately will show up as amino acid in the protein. So I'm changing the promoter, not the gene. And the last one is the gene is the same, but I am changing the transcription factors. So if you have a transposable element landing on a gene's promoter, so here's, I was trying to see, do you know that the promoter is the regulatory element? So that's B1. And here, as the example I gave you earlier, the gene encodes an enzyme, and maybe the enzyme will work well, maybe it won't. So that would be the coding sequence of a gene. And in this case, the gene would be an enzyme. It's not always, but very often is the case. And then you're like, oh, by exclusion, C is 3. They should say 3. So let's go over what this is showing. So this is sunflower that in the wild type, it's pink. And with this mutation here, so this thing here, if you took cell bio, you know, that's a construct. So like the colon. If you didn't know, now you do. So this means that they took this DNA sequence and they used a promoter. So 35S is a promoter, and it's called a strong promoter. So meaning it recruits the transcription factors really well. So the mRNA, the RNA polymerase will then be attracted to that sequence. So this part is a promoter, and then this is the gene that they transformed these plants with. So here it's pink, and here is white, a little bit of pink. Okay, how does it become pink? It's when you express these anthocyanin genes. I only showed you one in the previous question. I love like flower colors because it's easy to understand and to see. But these are different genes that would be involved in the anthocyanin. And then, so here's the start of transcription, right? So the gene starts here, and then there's going to be different genes here. So this, you'd have like one DNA, and then for that, another DNA sequence for that. So these are all different genes. They're all involved in the pathway of making the pigment. And then here's upstream, that's the promoter. These blobs here, they are transcription factors. So that WD-40, that sounds like oil. Anyway, these are the transcription factors that bind this DNA sequence. Now, if this DNA sequence is changed, so if this promoter is changed, then these proteins may not bind as efficiently. Or there is also the opposite effect of, if you change this DNA sequence, maybe some protein that wouldn't bind very tightly now does. So in this case, it's when they bind, you get transcription of these genes. Now the bottom one was a flower that was transformed with that construct. So some gene that they introduced in the flower, and then that gene, so that appears over here. So let's see what's the difference between these two. You're like, okay, that's the same, that's the same. that guy and here, these are the same, but whoopsie, now you have this one, this gray one, that's what they injected in the plant. So, well, not really, but imagine they injected that. This is a new transcription factor that doesn't exist in the wild type. And what it does is it binds this DNA more tightly than that. And as a result, this gene here does not get transcribed. Apparently the other ones do. So the proanthocyanin biosynthesis, so all of these get transcribed. This gene here doesn't. So this, what I'm trying to show you is that the transcription has to do with the transcription factors bound to the promoter. So it's for this gene specifically, that transcription factor has to be in this complex. When it's replaced by something else, you don't transcribe it. So the gene, the expression depends on the sequence on the promoter, and also depends on the sequence and if the transcription factors are bound to the promoter to promote transcription of that gene.